Okay, yeah, so I'm going to try and articulate our collective vision, but basically, we're imagining a way using this architecture that we can assemble, in this case, proteins into more complex structures towards these molecular machines. And, we chose to focus on proteins in this case, but you could imagine doing it with a spiroligomer or nucleic acid system as well. And so just to sort of outline or reiterate what the envisioning of this architecture is you have a functionalized solid surface, this workpiece that is being used to assemble protein Lego blocks using a sequentially microfluidically controlled process. And so these protein blocks are designed in such a way so that there are domains that are going to non-covalently and covalently interact in a deterministic way to link these blocks so that as you microfluidically cycle your sequence, you're going to build up on the surface these larger and larger assemblies. And you can also imagine that additional inputs in the form of light or different solutes, in addition to the protein blocks, may be also added to the system during these steps to build up and eventually remove them from this workpiece. And so here's sort of an illustrative sketch. So what one important thing to point out is that these proteins, they need to be designed computationally and then synthesized. And then once you have everything ready, you're going to go ahead and carry out this process. We chose to focus mostly on the Lego blocks themselves and how they're going to start to join together. There is definitely a important part of this, which is the workpiece itself, what that surface is, how it's textured, its chemistry, and how that's going to be able to interface. We only had so much time to think through. So we're focusing on the proteins, the blocks and the domains on those blocks that allow them to assemble. But basically, with each additional step, you start to build up a structure. These blocks can also be, we envision them to be also not just the same protein, but different ones, and then eventually cut them away. So which parts of this exist? Well, an automated microfluidic device is a well developed piece of technology. There are solid supports for protein binding and assembly, but as I mentioned, new kinds of surfaces and chemistries may be needed to do this. We can also de novo design and manufacture recombinant proteins. However, what's really missing, or one of the main things that's missing, is a library of what these blocks are. And the domains within these proteins, which are going to facilitate those specific, reliable interactions so that you can start to build up these Legos. And so this is something that hasn't been fully explored. And in terms of the applications that we can imagine from being able to do this, so you can create multi-component arrays or superstructures of proteins or blocks towards some sort of Lego castle that could maybe just be a certain structure, but you can also think of functional machines that could be motors or actuators, primitive molecular machines. And then another point we wanted to make is that maybe what we're trying to do here is not to create that end functional device, but to basically leverage this approach to build up subcomponents that then could be inputs in some of these other architectures, such as the printer or these breadboard approaches. So what is the sort of minimum viable architecture that we're trying to achieve here? Well, just as sort of a benchmark to compare ourselves against, bacteria flagellum is composed of over 30 different distinct protein blocks. So this is quite a challenge, but it again, require a library of many different blocks with many different types of domains. And what's going to be necessary is that as you go through these sequences, as you build up on your workpiece, once you've assembled two blocks, that binding interaction needs to hold up as you go through the later steps. So when you design a protein in a domain, you're thinking not only about that sequential step from A to B, but also all the follow-on steps that are going to come after. And so that really is a huge challenge as we see it. And so the good news, and maybe, is that there is a large space of proteins structures to be explored using state-of-the-art computational tools, and so this is a frontier that has possibilities that are ripe for exploration. So I think that is the end of what we've got here.
Does anyone want to add anything from your group? Because you still have about a minute. Please give him the mic.
I think something just in general with all the themes of the different architectures that we've explored over the past today and yesterday is just as the last group pointed out, a lot of these pieces could be really complementary. And so just as Caleb was pointing out on the killer apps, that maybe this could be used to form subcomponents of the molecular printer idea. And I think there's a lot of feedback between these approaches, and it shouldn't be thought of as like the one perfect architecture. I really liked yesterday the discussion from William about the idea of templating tips for probe-based synthesis using biomolecules, and I think that's one of the things that I just find that most encouraging from today's discussions.
Wonderful.
I think we spent some time, I think the word creative tension was used, trying to decide whether covalent interactions versus non-covalent interaction of the Lego pieces, and both could be mix and match. I tend to favor covalent interactions. I brought up the fact that there is this very unique system called SpyTag SpyCatcher, where two domains on two different proteins will form a covalent attachment. So it's possible to build that into the protein designs, but certainly then someone else pointed out that bacterial flagellum, the proteins have non-covalent interactions, and those seem to be fairly strong. So we're a little bit agnostic there, again, but it's as Caleb pointed out, the real challenge is in the design of those, the computational design of those Lego pieces.
Cool. Thank you. Now we have a bit more time for questions, comments. Anyone? Wow, we already have a few. Okay, we're just going to go through this row.
Yeah, that's pretty cool. Thank you. I guess in terms of like what's missing, I think if you go back a few slides, you were saying like so. I just want to comment here that like the design of a library of protein blocks that assemble in that way is already done. It's going to be published pretty soon. What we don't have is like the sequential assembly line that you describe here. So that would be pretty cool to do.
So the blocks exist, but do you know how they interact with other blocks?
Yeah, so we can make blocks and then we can design interfaces between them so that they just bind to each other in very specific ways. So that's done. But then the problem also is like when you have this like assembly step and you use either covalent and non-covalent interactions, how would you go about designing things that have symmetry, right? So if you want to make machines, for example, you need things that move relative to each other, right? And so I'm not clear how you would do that. Like you cite the bacterial flagellum, it's like, it's like super high cyclic symmetries and like multiple components that have this kind of like degrees of freedom. How would you assemble that?
Does anyone want to take that?
You're asking how we would impart symmetry or asymmetry?
Yeah, for example, if you have like 30 building blocks that make a ring, how would you assemble that into your into this like step-by-step assembly protocol that you describe here?
Well, again, I think we decided that you'd have to design each part such that as you flowed it in, it would only interact in one specific way. We did discuss whether we needed deblocking reactions or uncaging reactions. So maybe you need to protect one face, bring in the molecule, let it interact, deprotect it, and then bring in the next building block.
Right,
So,
but for example, if you have like a C30 symmetry in the bacterial flagellum, it's going to assemble in solution. How are you going to, you'd have to have, if you want to have the symmetry, then you, so you'd have essentially to have symmetrized the whole C30 complex for that to be assembled step by step.
well, so I think to prevent it from aggregating in solution, you would probably have to protect one domain such that it couldn't interact until you wanted it to.
Yeah, I
Can
have
I
to
just say,
draw
can the
it.
blocks be multivalent? Like,
Well, if you have symmetry, then the building blocks are gonna. I mean, I have to be.
Go for it, Chris.
Yeah, sorry.
I think this is also a great place to again point out just the ways in which all these different approaches can be really complementary. And yeah, but like I think as our discussion of a flagellum was just inspiration, but stands to reason like a lot of the work that like you've pioneered, there are perhaps better approaches for making something very symmetric. And yeah, we can certainly think about like how this platform, what are those like asymmetric pieces of other machines we want to build where this could excel, where we're not trying to reinvent the wheel, where there may be better approaches for making the symmetric type molecules already exist. I think we really need to think critically what the molecules that we can't make, where can this platform excel?
I totally agree. I think it's very exciting. I was just commenting on the fact that designing symmetry with that approach is really hard. Yeah.
I think my question was kind of largely answered. So.
Over here, thank you.
So something on the lines of what has been asked before, but are you? So I was wondering. So because you were showing the Tetris, the Lego shaped Tetris, which was getting built up, how? So my only thought was, did you think about what happens if it doesn't attach like you want? It attaches some way other, because if you, let's say you want to attach it by a covalent bond like that, but then there are other interactions which are non-covalent, which can attach it in the different orientation, then how would you stop that from happening?
Well, that's up to the computational design people.
Push the can down the road. Okay, did you have a question?
Yeah, I just have a comment. A lot of this stuff is similar and could build upon the work on megamolecules from the Merxish group, who have already done a lot of this stuff using spycatcher and snap tags and all that stuff, and it's really, really similar to this.
Well, you already had a helpful comment. Great, you preempted one of the discussion prompts. Any final bits and pieces? Okay, well then maybe let's go back to the doc and see if we can fill anything in here from the discussion prompts. Did you describe any like next steps or any in particular things that you would do with a development grant that is different from snacks?
I think we also have that one blank,
You had
unfortunately.
snacks. Okay. Snacks. More snacks. Feedback received. Okay. Anyone else want to say anything particular that they thought was particularly amazing or particularly missing from this architecture that we haven't tackled yet? Anyone else who should be working on this, who they should be contacting?
Did you do computations or estimates of how many different tags are needed, like mating surfaces, protection, deprotection? That might be an interesting number to find out.
Yes.
Okay, anything else? No? I think we're right on time. So thank you so much. Wonderful.